Protein structure. Shape is critical to the function of a protein. A protein's shape depends on 4 levels of structure: primary, secondary, tertiary, and quaternary. Proteins come in all different shapes and sizes. The smallest functional peptide is a tripeptide, glutathione. The largest known human protein, titin, has 34,350 amino acids. The diagram shows a polypeptide chain of alanines, an amino acid. Some hydrogens are not shown. Green is carbon, blue is nitrogen, red is oxygen, and white is hydrogen. The primary structure of a protein is the sequence of amino acids in the polypeptide chain. Each protein consists of a unique amino acid sequence. The order is important because it determines how they fold. The interactions of amino acids with their environment and with one another determines protein shape. For example, each tetrapeptide below would have a different shape in a protein, even though they have the same amino acids. Secondary structure is the result of hydrogen bond formation between amine and carbonyl groups of the polypeptide backbone. Depending on where the groups are relative to one another, the secondary structure takes the shape of an alpha helix, shown to the right, or a beta pleated sheet, shown on the next slide. Note, R groups do not play a role in secondary structure. The hydrogen bonds, indicated by dotted lines, between the backbone groups maintain these shapes. Tertiary structure is the overall 3D shape of the polypeptide, the arrangement of alpha helices and beta pleated sheets in space. It results from interactions of R groups with one another in their environment. R groups cluster or repel due to hydrophobic and hydrophilic interactions. They attract and repel one another due to charge interactions. Some proteins have quaternary structure. Quaternary structure consists of more than one polypeptide chain bound together. Tertiary and quaternary structure are often stabilized by disulfide bridges, covalent bonds between the sulfhydryl groups of 2 cysteines. IgG2 antibodies are made up of 4 polypeptides. They're stabilized by hydrophobic interactions, hydrogen bonds, charge interactions, and 2 disulfide bridges indicated by the arrows. Here's a summary. Complete protein structure: primary, peptide bonds between amino acids; secondary, hydrogen bonds between NH and CO in the backbone; tertiary, hydrophobic interactions, charge interactions, and disulfide bridges between R groups and with the environment; and quaternary, multiple polypeptides combine. Here is a table summary showing all of the structures and notes for every level. The primary sequence of amino acids and their interactions with their environment determine how a protein folds. Shape determines function. Each sequence of amino acids folds in a different way as each amino acid in the chain interacts with water, lipids, and the other amino acids in a unique way. For instance, upon contacting water, a protein could form a groove with an arrangement of electrons that functions as a binding site for other molecules. HIV-1 protease, a protein that cuts other proteins in its cleft. Changing an amino acid or the sequence of amino acids in a protein changes its primary structure and can affect its overall structure and ability to function. Sickle cell disease is an example of a single amino acid defect that changes the shape of a protein, causing a disease. Hemoglobin carries oxygen in red blood cells to bring to the rest of the cells in the body. Replacing one glutamate amino acid with a valine amino acid causes a change in the overall shape of the hemoglobin protein. Sickle cell hemoglobin clumps together, changes the shape of red blood cells, and leads to problems with the circulatory system. Changes in a protein's environment can change the way it folds and bends. Structural changes may inhibit or enable a protein's function. Extreme changes in heat, pH, and salinity can cause proteins to denature, which means they unfold permanently and completely lose their functionality. This egg's proteins have undergone denaturation and loss of solubility caused by extreme heat during the cooking process. Biochemical reactions have stopped and the egg will not return to its previous state or develop. Proteins have thousands of different roles in an organism. Keratin is the main structural protein in hair, skin, and nails. Aquaporin forms channels for water through cell membranes. Hemoglobin transports oxygen and carbon dioxide in the bloodstream. And green fluorescent protein is a signaling molecule for jellyfish. 7 common categories of protein function are shown. Structural, hair, cell cytoskeleton. Contractile, as part of muscle and other motile cells. Storage, sources of amino acids or ions. Defense, antibodies, blood cell receptors. Transport, hemoglobin, membrane channels. Signaling, hormones, membrane receptors. Enzymatic, regulates speeds of reactions. And machines, motor protein moves organelles, microtubules move chromosomes.